So moving on to chapter 15, we'd like to talk about how things move from one compartment to another, how things get into the endoplasmic reticulum, how they move from the endoplasmic reticulum to the Golgi, how they move from the Golgi to maybe exocytosis or a lysosome, or how proteins that are made in the cytosol enter into the mitochondria or the nucleus. And so this idea of intracellular trafficking is really at the core of cell biology. Cell biology has kind of a pantheon of gods, and one of the leading cell biologists at the beginning of the 20th century was a guy named Blobel. He just passed away last year, but he really allowed us to understand how things move from one compartment to another using pulse chase experiments and moving tags around on proteins to see where they ended up with different signal sequences. So he's the person who came up with this idea of proteins have an inherent signal sequence. And the way that he did that was he took a protein that was normally resident in the endoplasmic reticulum, and he took off a portion of that protein and attached it to a protein that was normally resident in the cytosol. And what he found was that proteins that were normally resident in the cytosol but had the endoplasmic reticulum signal sequence ended up in the ER. And proteins that were normally in the ER, when they had their signal sequence removed, would end up in the cytosol. So this idea that there were signal sequences led to a greater understanding of intracellular trafficking. So the consensus signal sequences for many different compartments has been worked out. So there's a signal sequence for entering into the ER, and that is at the N-terminus. If you have a chain of, say, like 10 or 15 hydrophobic amino acids, it just makes sense that as that protein is getting made in the cytosol, because all protein production starts in the cytosol, then the growing polypeptide chain, because of this hydrophobic nature of the amino acids that are coming out of the ribosome, it would not like to be in the cytosol because it's so hydrophobic, and the cytosol is of course all water, and so the signal sequence is going to be energetically favorable for binding to a signal recognition particle to cover up the hydrophobic region. And then that signal recognition particle would then target this protein to the ER. And so any protein that is going to be resident in any of the compartments or in any of the membranes is going to have some sort of signal sequence. So once a protein is recognized as being either supposed to be in the ER, supposed to be in the Golgi, supposed to be in the lysosome, or in any of the membranes, or if the protein is supposed to be secreted from the cell, exported from the cell, then it is going to have this short hydrophobic region at the N-terminus. Now, that's only going to target it to the ER. That's not going to guarantee that it stays in the ER. there's a completely separate sequence that causes retention in the ER, and that's at the C terminus. On the other hand, if it gets imported into the ER and then doesn't have that retention in the ER signal sequence, then it will get moved to the Golgi and the lysosome, because the default pathway is secretion from the cell. And so then there are other signal sequences, proteins that get made in the cytosol, but have a consensus sequence that causes them to be imported into the mitochondria, or a signal sequence that targets them to the nucleus or the peroxisome. And so these signal sequences and the discovery of them is really essential for our understanding of how things end up in the right place in the cell. So once that signal sequence is recognized, what is the sequence of events? So like I said, all protein production starts in the cytosol. So the mRNA gets exported from the nucleus and recruits the small ribosomal subunit, which comes preloaded with a tRNA for methionine, and so the small ribosomal subunit scans down the RNA until it finds the AUG start codon. That causes it to recruit the large ribosomal subunit, and then translation continues. And so then you'll have a growing polypeptide chain coming out of the ribosome. And if it does have this ER signal sequence, this short section of hydrophobic amino acids on its N-terminus, then it will get recruited down to the ER membrane. So the way that happens is that a signal recognition particle, a chaperone protein, binds to this signal recognition particle and stops translation for a period of time until it gets recruited down to the ER. The signal recognition particle binds to the signal recognition particle receptor and migrates to a translocation channel, the ch15, sec standing for secretion from where it was discovered in yeast. So the ch15 is the translocation channel, and the signal recognition particle receptor hands off the ribosome and the growing polypeptide chain to the ch15 translocation channel, where the signal sequence binds to the inside of the channel, and then protein translation continues. It starts up again, and so now you've got the growing polypeptide chain going into the lumen of the ER, and it's going to continue going into the lumen of the ER until it's completed, at which time another protein, the ch51, will come and cut off the signal sequence. So the signal peptidase cuts off the portion that is bound to the inside of the channel. And so now you've got the entire protein has been made and threaded into the lumen of the ER. The signal sequence will just drift off into the membrane and be degraded, and the translocation channel goes back to ready to receive another ER resident protein, or a protein that's going to be somewhere in the endomembrane system or secreted from the cell. So there are proteins that span the membrane and how they span the membrane is after the signals, or after the protein has been recruited down to the translocation channel, there will be a second hydrophobic region, and that hydrophobic region is going to be, some alpha helix that is going to be the right size and perfect for spanning the membrane. And so when that sequence gets threaded through the translocation channel, that will stop translocation. And so then the growing polypeptide chain will drift out into the membrane, and translation continues, but now it's going to be on the cytosolic side. So that's how we get a single-pass transmembrane protein. And you'll notice here that there is a defined orientation, because the first part that was moved through the translocation channel is the N-terminus. The N-terminus would be luminal or external to the cell later on. Now, not all proteins have this orientation of N-terminus on the inside of the ER and C-terminus on the cytosolic side. And so we would have to have a different sequence of events for single-pass transmembrane proteins that go the other direction.